as you know, today I'm going to talk about the metabolism of the amino acids. In the previous semester, you will be familiar with some important biomolecules, the structure of some biomolecules, and as you know, one of the important biomolecules which has several and diverse functions are proteins. And they compose of some building block, and they are amino acids. So in this session, I will start with the digestion of the protein, because, for example, if you ingested a meal containing protein, at first I'm going to start what happened to this macromolecule, what happened to this protein, and finally how we can produce energy, or how we can produce some important compound from the protein, because protein not only have, for example, enzyme function, and also not only they provide, for example, some function for the immunity, I mean immunoglobulin, and also, for example, they are some carrier that they transfer some biomolecules. So not only these functions are regarded for the proteins, but also they provide some precursors. I mean the amino acid for many, for example, hormones. Because, for example, serotonin is a hormone that derives from a specific amino acid. And also we have a histamine in an allergic response. Our body, I mean the specific cells of the body, for example, mast cells produce a histamine. So not only they produce a building block of amino acids for biosynthesis of new proteins, but also they participate in a production of some compounds. About the reference, lipid code I think is the best reference for you, and I put the folder and the name is the metabolism of amino acid. I put the slides of 2 sessions and also the text of the book. If you want to get more information, you can refer. But as you know, lipid code may include extra information, but I mentioned this point, these are just for more information, and you don't supposed to memorize the extra information. So please focus just on a slide. So if you have any problem in understanding the slides, you can refer to the book. About the outline and about the purpose of this session, at first I will have an overview about the amino acid metabolism, and I will define what is the amino acid pool and what is the source and fate of the amino acid. And then I will introduce a pathway that they participate in protein turnover and digestion and absorption of the dietary proteins and transport of amino acids in the cells. And then I will continue with the catabolism. We have some reaction. These are general reactions for all kind of amino acid, because the first step is a removal of the nitrogen, removal of the amine group by transamination or deamination, and finally they exerted into the urine in the urea cycle that happens inside the liver. And in the next session I will describe the continue of the metabolism. In this slide you can observe the overview of amino acid metabolism. Okay. Intracellular, you know, we have 2 sources. Some of them is intracellular, so normally our body produces proteins. So we have some intracellular proteins, and also sometimes we ingest the protein from the diet plan. Ingest the protein from the diet. So no matter if this is the intracellular or extracellular or in the diet, all of them should be converted to their building blocks, I mean the amino acid, okay? So the first step is converting to the amino acid. In the next slide, I will introduce how we can reach to this step. Whenever we produce these building blocks, we have another specific catabolism. The first and the obligatory step during the metabolism of amino acid is a removal of the ammonium. Do you remember the structure of amino acid? They contain actually this is a structure of the amino acid, this is the general structure. They contain the H, they contain the NH3, COO, and also R. Okay? I mean, the first step of the catabolism of the amino acid is the removal of the amino group. So, in this session, I'm going to talk about the removal of this ammonia, and finally it exerted into the urine as a urea. This is a disposal form of the ammonia. Okay, and what is the name of remaining structure? This is the alpha keto acids, because if you remember, inside the structure of amino acid we have a carbon alpha. Okay, so, and also because of the presence of the carboxyl group, this remaining structure known as the alpha keto acid. Okay, so whenever the amine group is removed from the structure of amino acid, the alpha keto acid, and in the other words, carbon skeleton is remained. Okay, I'm going to talk about this part in the second session. And now I'm going to talk about what is the procedure to reach to the amino acid, and finally how we can remove it, this ammonia, and how we can dispose in a urine. As I mentioned, we have an amino acid pool. What does it mean? It means that in a healthy, well-fed individual, the input and output of the amino acid are in balance. It means that the amino acid pool that is present throughout the body, for example, maybe inside the cells, maybe in the blood, in the extracellular fluid, we have the total amount of amino acid everywhere. If you consider all this amount of amino acid, the name is amino acid pool, okay? And the amino acid pool is small, okay, compared to the amount of total protein. The amount of the building block of protein is very small. But generally, in a healthy, well-fed individual, there is a balance between input of amino acid, maybe you ingest from diet, or maybe our body cuts the protein into the amino acid and providing new amino acid for the biosynthesis of the protein, for biosynthesis, many hormones, and etc. So what is important here, we should know that there is a balance between input and output of the amino acid pool. Okay, how we can provide and how we can actually, what is the source of amino acid pool? I mean, I mentioned this point, we have a balance between input and about the output. What are the source of the input of amino acid? Amino acid may be provided by the degradation of the proteins. Okay, finally, proteins, for example, if you have unneeded protein, or sometimes you have abnormal protein, finally they distinguish by the body and they cut and digest to the amino acids. So one of the source of this pool is a degradation of the protein that present inside the body. And also, maybe you ingest some amino acid, some free amino acid, or some proteins that finally inside the gastrointestinal, finally they convert to the amino acid. And the last source is synthesized from non-essential amino acid. Do you remember we can classify amino acid according to the nutritional aspect? Essential and non-essential. So essential amino acid should be provided and supplied in the diet, but non-essential amino acid have an intermediate and have a precursor inside the body. So our body, in a normal situation, yes, they synthesize these non-essential amino acid. What about the output of amino acid, and actually how we can consume and deplete these amino acid pool? It's very clear. You can use amino acid for the protein synthesis. You bind all of them together by the peptide bond and form a functional protein. And also, Amino acid consumed as a precursor of some small molecules. And at the end of the second session, I will introduce some important compound like, for example, histamine, nitric oxide, catecholamine like epinephrine, norepinephrine. All of these molecules contain nitrogen, and most of them are derived from amino acids. So this is one of the ways to use and consume amino acid. And finally, they convert to the glucose, glycogen, fatty acids, and ketone bodies, and also CO2 and H2O. And this pathway is very important to the producing of energy. You know, for example, if you, I think that you have a carbohydrates metabolism section. During the carbohydrates metabolism, we have a one pathway, gluconeogenesis. During the gluconeogenesis, some precursor convert to the final they produce glucose. Okay, for example, one of them is an amino acid like alanine. Okay, it is considered as a glucogenic amino acid. Okay, these are the review of the fate and source of the amino acid. And about the protein turnover, you know, it means that protein are not immortal. We have a half-life for proteins, but we have a different half-life. But normally, whenever they reach to the end of the life of protein, they digested by the body. And sometimes they produce some compounds, and sometimes they exerted into the urine as other metabolites, okay? And sometimes they store a small amount as a source of the amino acid. We have a little storage of amino acid. So most of the proteins that constantly be synthesized and then degraded, it means that again, we have a balance between rate of biosynthesis and also rate of degradation of the protein. So the total amount of the protein is constant. How we can remove it actually, and sometimes not only whenever the protein reaches the end of the life, but also sometimes we have abnormal protein like mutated protein. Our body distinguishes them by a specific pathway. In the next slide, I will introduce this pathway. And sometimes, for example, we have an unneeded, okay? So it means that our body produce and synthesize more than your requirement. So your body should degrade these extra amounts and excess amount of the protein. How we can degrade the protein? According to the classification, at first I'm going to classify according to the half-life of the proteins. Because the rate of protein turnover of the protein is different. Sometimes they are short-lived protein, okay? And the half-life measured in, for example, minutes to hours. For example, regulatory protein and also misfold protein. You know, because if you have a high half-life for the regulatory, maybe they disrupt the metabolism, okay? They produce and immediately function and regulate the enzymatic reaction, and immediately they remove from the body to control their function, okay? So they have a half-life measured in minutes and hours. These are regulatory proteins. And also we have some long-lived protein, and majority of the protein are belong to this group. For example, like immunoglobulin and many many functional protein like enzyme are belong to this group. And the half-life measured in days and weeks. Okay? And we have a structural proteins. A structural protein, you remember the protein station, we have some structural like collagen, like elastin. They don't have any specific function like hemoglobin to transfer the oxygen, I don't know, myoglobin to reserve the oxygen. Because they provide a structural function, so they have a very long half-lives, okay? They stay for a long time. So this is very important. What is the example questions? For example, maybe I ask you, which of the following protein has a short-lived half-life? Has a very short half-life. And for example, maybe I ask you, which of the protein they have a half-life measure in months or years? You should choose the structural proteins, and the collagen is an example of structural proteins. Okay, so now we have it, for example, short-lived or, for example, long-lived. And finally, we reach to the point that we have to degrade the protein. What are the mechanisms, for example, for the removal of the abnormal, unneeded, or this kind of protein? What is the procedure? We have two systems, enzymatic systems, that are responsible for the degradation of the proteins. One of them requires ATP, another system actually is an ATP independent degradation system. And this is a non-selective pathway. But here we have a selective pathway. In this pathway, ATP-dependent ubiquitin proteasome system of the cytosol, some damage, some abnormal protein, a non-functional protein, or some short-lived protein degraded by the endogenous proteins. In the next slide, I will describe this procedure. You should know that in this procedure, they require the source of energy in the form of ATP. But in this way, this is the ATP independent. There is no necessity to the source of energy. And most of the extracellular proteins, and such as plasma protein, they taken into the cells by the endocytosis. Are you familiar with the endocytosis? Yes. They envelop the extracellular protein, and sometimes we have some specific receptors. If you remember, for example, this is a cell, and we have some specific receptors that they can determine, for example, abnormal protein, and finally they ingest that protein, they envelop the protein, and they digest the protein, okay, by some organelles like lysosome, okay, they hydrolyze by the acidic hydrolyze that are located inside the lysosome in some vesicles. This is a general pathway. So most of the protein, for example, that are like plasma protein and extracellular protein are degraded by this pathway. But what is the ATP dependent? What is the ubiquitin proteasome? This is a pathway that one of the important pathway for degradation of the protein. As you can see here, assume that this is abnormal protein, or this is a short-lived protein, for example, or this is a mutated protein, this blue part, or cellular protein. How we can send the signal to our protein, to our body, that is an abnormal protein, or that is a non-functional protein, and how we can tell to our body, please degrade this protein. As you can see here, we have a glycoprotein, and these are a small globular protein named as a ubiquitin. Okay, they use for tag and for actually make a specific structure. I mean that they bind to the non-functional, and also they bind to the abnormal protein, and they tag the protein. Whenever these tags are attached to the protein, it sends the signal, okay? Like, for example, the red light, okay? For example, you have some proteins, and this is abnormal, okay? Our body tagged with the ubiquitin, and this ubiquitin function as a red light, okay? This is abnormal protein, this is abnormal protein. Please digest this tagged protein, okay? How?
Antibodies is the same mechanism of antibody?
Yes, these are different, but in some way they are similar, okay? So whenever our body tags the abnormal protein, This tag protein distinguished by the large barrel-shaped complex known as a protosome. Okay? So this large barrel-shaped complex takes the tagged protein and by the ATP dependent, like here, as you can see here, for the binding of ubiquitin, we require ATP. So this system is an ATP dependent system. So you should know that adding ubiquitin requires ATP. After adding ubiquitin and taken by the protosome, finally inside this barrel, the protein cut into the small fragment. Okay, like for example, decapeptide, tripeptide, and tetrapeptide, and finally by the some proteases, they are non-specific, they produce a free amino acid. So from the large protein, finally we reach the amino acid. And also, as you can see here, ubiquitin is recycled, so you can use it for several times. Okay, so this is the red line with a high half-life. It can use for many cycles. Okay, it can tag many proteins. It can recycle. Okay. In this slide, I'm going to show what happens whenever you ingest the protein. Okay. Do you have any information about the digestion of the protein? Which organs are participating?
Protease dissolves proteins.
At first, let's start with the name of organs.
Stomach, small
stomach.
intestine.
Pancreas. Excellent. So we require this organ to have a fully catabolism of the proteins. So whenever you ingest the proteins, if you remember about the catabolism of the carbohydrate, we have an amylase inside the salivary. So the catabolism of the carbohydrate starts and begins inside the mouth. What about the protein? It starts inside the stomach. Okay? So we have a different kind of the cells. We have a parietal cells and also we have a chief cells. Do you remember the difference? We have acid producing cells and we have enzyme producing cells. So chief cells produce the enzyme, okay? And parietal cells produce the HCl, okay? So following the entering of the protein inside the stomach, our stomach produces acid and pepsin. And inside the stomach, they digest and cut and break down to the shorter fragments of the proteins. But following the so the gastric secretion like pepsin, and you know that these are not active protein. Most of the digestive enzymes, especially the enzyme that participate in the digestion of the proteins, produced in a proenzyme. For example, at first our body produces pepsinogen, okay? And finally it converts to the pepsin. So pepsin is an active enzyme. So it releases some peptide, as you can see here, peptide and a few amounts of free amino acid. Okay, on entering to the small intestine, we have a secretion of the pancreas. We have some pancreatic enzyme, and also we have an alkaline environment by the secretion of the bicarbonate. And also the enzyme, we have entropeptidase or endopeptidase and exopeptidase, and in the next slide I will introduce some of them. By the help of the bicarbonate and this enzyme, finally when we have a short, and for example, as I mentioned, maybe you have a 30 amino acid, maybe you have a 10 amino acid that bind together, and finally they gradually, they cut to the amino acid, free amino acid. And also we have some hormones, okay? we have some hormones that secreted from a specific gland, okay? But about the gastrointestinal, we also we have some site to produce some gastrointestinal tract hormones like cholecystokinin and secretin. These are two hormones that participate in a release and activation of the zygogen, especially of the pancreas. And we have activation of the trypsinogen by the entropeptidase, and formerly it's called enterokinase. In the next slide, I will show them. Okay, this is a review. For example, if you intake proteins finally by the help of the stomach and pancreas and small intestine, we finally reach to the amino acid. I think yesterday I asked you why we require different type of enzyme to digest the proteins, and you mentioned that because of the specificity of this enzyme, they are required in a different type. Some of them, for example, hydrolyze the peptide bond, that the carbonyl of peptide bond, for example, contributed by the lysine and arginine, like a trypsin. And some of them cut the side of, for example, bulky amino acid, like tryptophan, tyrosine, phenylalanine. And so, as you can see here, we have a specific site for many enzymes. Please pay attention to this point. As I mentioned, most of the digestive enzymes like trypsinogen, chymotrypsinogen are produced in an inactive form, okay? So we require another enzyme to activate them. As you can see here, what is the activator? Which enzyme is the activator?
Entropeptidase.
Entropeptidase is the activator of the trypsin. Whenever our body produces an active trypsin, so this trypsin can catalyze the activation of the other enzyme, like chymotrypsin, elastase, and carboxypeptidase A and B. So if I ask you, what is the general activator of the digestive enzyme? The name is trypsin. Please pay attention to the blue arrows, okay? Like anything else.
Is it, should we also memorize, for example,
The site?
And I meant, for example, like trypsin, it acts on which amino acid?
The site is not important because I think it's difficult to memorize because I make a mistake.
Right now, the first, the trypsinogen, right? Trypsinogen. Then by enterobutase, the enterobutase
we have a cascade, okay? At the beginning of the cascade, trypsinogen convert to the trypsin. Why which enzyme? Entropeptidase. And this trypsin start the following reaction by the activation of the elastase, carboxypeptidase, okay? This is a general activator. At first, what is the trigger of this cascade? Activation of the trypsin. Whenever trypsin is produced, it can continue this procedure. It's like a cascade of the digestion of the proteins. No, the size is not important. You don't suppose.
That domino effect, the cascade,
Gastric, what do you mean?
I mean the domino effect, like.
The active side of enzyme is
exactly. Like a domino effect, for example, if they are here, if you start, for example, this is a trypsin. Trypsin starts an activation. Excellent, and thanks for your good example. Exactly like this. Okay, like other metabolism, like for example, metabolism of the lipids, metabolism of the carbohydrate, sometimes we have some abnormalities and deficiencies regarding to the metabolism of the proteins. For example, as you know, I mentioned that for the complete degradation of the proteins, we require the secretion of the pancreas. So it's very clear that whenever you have some problem inside your pancreas, you have inflammation, I don't know, you remove this gland by the surgery, or maybe, for example, you have a cystic fibrosis, any disease that can affect the function of the pancreas can affect the digestion of the protein. So it's regarded as an abnormality. And also, one of the important examples of the abnormality of the protein digestion is a celiac disease. Celiac disease, these patients actually are very sensitive to the gluten. Maybe you encounter with some products and they mention, for example, this product is a gluten-free. Okay, because gluten is a kind of protein and it can be found in, for example, in a wheat, in a rye, in a barley, and maybe they make some sensitivity in some people. So you should prescribe them. Please use some products that are gluten-free, because our body, by mistake, produces some antibodies, and so they result in some clinical manifestation. So, in a celiac disease, we have an abnormal protein digestion. So we should limit the intake of the protein for these patients. Okay, we digest the protein, Stella. We digest the protein, and finally we reach to the small peptide like di or tripeptide or free amino acid inside the intestine. So I'm going to talk about what happened to these free amino acids, okay? And how our enterocytes are the cells of the intestine, how they can absorb and take these free amino acids or these small peptide. As you can see here, this is, for example, intestinal lumen, okay? This is intestinal lumen and this is an apical membrane. As you can see here, we have a sodium link secondary transporter at the apical membrane to take up the free amino acid. When we say secondary transporter, it means that desired molecule enter with another component. This is a secondary.
Replacement between the component inside the membrane and the outside the membrane.
Yes, and sometimes this is exchange. For example, sodium enter, potassium exit the cell. These are exchanger. These are some transporter example. For amino acids, so the presence for the it means that for the absorption of amino acids, sodium have a specific role. Why? Because we have a transporter that is sodium link. So whenever they are taken into the enterocyte, finally by the facilitated diffusion, they reach to the portal vein. And what happened here also, we have some transporter. Maybe the protein digested to the free amino acid, so we have some specific free amino acid transporter. But sometimes we have, for example, tri or dipeptide. They cannot taken up by this transporter. So how can I take up? By another specific system. And these are the proton link transporter, and whenever they enter inside the cells by the help of enzyme, finally they degrade to the amino acid. So please consider this point, only free amino acid are present in a portal vein. So there is no dipeptide, intact protein, tripeptide, all of them should be digested to the amino acid, only one amino acid. Okay.
After I digest the tab and enter the cell, where's where will we go after that?
After that, we have another phase. I will gradually describe it, okay? At first, I will start with the digestion of the protein. by the stomach, by the intestine, by the secretion of the pancreas, and they finally convert to the free amino acid and maybe di or tripeptide. They reach to the enterocyte and they convert to the portal vein and reach the bloodstream. Now what happened to these free amino acids? This is your question.
Now, my question, this is the time. After entering to the cell of
In this
intestinal cell,
peptide,
yes.
okay?
After entering, where is degraded here?
Yes,
And
by
same
this
cell?
enzyme. This is it. This, please consider this point. Inside the enterocyte, these are non-specific peptidase. No matter what is the sequence of amino acid, this is arginine, serine, lysine, these are the general and non-specific enzymes that are responsible for the digestion of the amino acids, peptide. But small fragments of peptide, okay?
So after-
Not large.
It's turned to amino acid and to
Yes, yes. Whenever they convert to the free amino acid, they send to the portal vein and they reach to the bloodstream. Okay, how we can transport the amino acid? Okay, I'm going actually to please follow me step by step. Okay, proteins digest to the free amino acid. Free amino acid enter into the enterocyte. If they are tripeptide, finally they degrade to the free amino acid. And this free amino acid reach to the portal vein and reach to the bloodstream. So they actually should send to the other part of the body. Okay? So we have some transport system for the amino acid. You should consider at this point the concentration of the amino acid, I mean free amino acid, in the extracellular fluid is significantly lower than inside of the cells. Okay, the concentration is very low. For example, this is cells. As you can see here, the concentration inside the cells actually is very higher than extracellular. So we have an active transport to transfer amino acid, okay, inside the cells. So it's very clear. Are you familiar with the active transport and passive? Whenever I want to send, for example, molecule from the high concentration to the low concentration, is it required to use the energy?
No.
No. What about amino acids? These are amino acids. Because as I mentioned, these are more concentrated and we have a dense concentration of the amino acid inside because I want to send these few amino acids, so I have to use energy. Okay? So we have some active transport system. Where are the site of this active transport system? In a small intestine and a kidney. Proximal tubules of the kidney. So whenever our body digests the protein and reach the free amino acid, they send inside the bloodstream and they reach to the small intestine sometimes and proximal tubules of the kidney, and they taken up by these organs. We have some disorder regarding to these transporters.
Why would we send proteins from low concentration to high concentration?
Because our body didn't want to lose even a small amino acid. Okay? Because if, for example, this is a lumen, okay? This is a diet. Finally convert to the amino acid, okay? We have some transporter at the apical membrane. We have a sodium link, okay? Do you remember? And also we have a bloodstream, okay? I'm talking about the site of bloodstream because our body wants to take even a small amount of amino acids, okay? Because our body is very stingy. It wants actually to hold any precursor, like amino acid because they are very vital for our body. So it's very logical to use the energy to take off these few amount of free amino acids. Okay? But sometimes we have some disorder regarding to this transport system. As I mentioned, these transport systems are located in two sites. One of them is a small intestine, another one is a kidney, proximal tubules of the kidney. One of these systems is responsible to uptake a specific amino acid like cysteine and some dibasic amino acid like ornithine, arginine, and lysine. With the abbreviation C is cysteine, ornithine, arginine, and lysine col. So this transport, for example, known as a col. Okay? And it participates, for example, in the kidney absorption of the amino acid. What is this cystinuria? When we say urea, what does it mean?
It means excreting through urine.
What about emia?
Emia, when there's not enough in blood.
Excellent. So about cystinuria, it means that this transporter cannot absorb the amino acid properly. So cysteine, actually most of the amino acid like cysteine and also dibasic amino acid secreted in the urine. Because they are not very soluble, they accumulate and precipitate inside the kidney. And they form a kidney stone. Okay? So some abnormality regarding to the catabolism and metabolism of the amino acid can result in a kidney stone. Because we have are familiar with the kidney, different kind of kidney stones.
Yes.
We have a different kind. For example, if you take a large amount, excess amount of the vitamin C, because vitamin C finally convert to the oxalate, it can form an oxalate kidney stone. Okay, so we have a different kind of kidney stone. And what happened? They obstruct the duct of the urinary tract and they form many manifestations. About the heart knob disease, do you remember the vitamin session? I introduced this disorder. This is the heart knob disease, and we have some defect in the transport of the like tryptophan. Okay, tryptophan is the important, and actually this is the essential amino acid, and this is vital for many aspects. One of them is the precursor of niacin. Niacin is a vitamin B3. B9 is a folic acid. Okay? If you remember, we have some deficiency. For example, about the deficiency of vitamin B1 or thiamine, we have a very disorder disease. In a thiamine, niacin, vitamin B3, we have a Plager disease. Okay? Why? Because in Plager disease, there is no enough amount of vitamin B3. One of the reasons of this disorder is the lack of precursor of B3. Okay? Because during the normal metabolism, tryptophan as an essential amino acid produces vitamin B3. In a heart knob disease, because our body cannot absorb this precursor, so we have a deficiency of these amino acids. So what would be the result? The deficiency of vitamin B3. And what is the name of the disease? Plager. Okay? So this is another example of the disorder of the metabolism of the amino acids. Okay, I reached to these points. Proteins digest to the small peptide and finally free amino acids. Free amino acids taken up by the intestine from the luminal surface and they reach to the blood, and maybe some of them resolve from the kidney. Okay, sometimes this amino acid hold intact. For example, as a precursor of some proteins, they have a function as a building block of the proteins. But sometimes our body wants to produce the energy, produces some precursors from the amino acid. So we have to digest and cut the structure of the amino acid. Now I'm going to talk about the catabolism and the fate of the amino acid. About the catabolism, we have a common reaction, removing of the alpha amino group, and what was the remaining structure? Alpha keto acid or carbon skeleton. And this carbon skeleton contains specific reactions. This is the overview of the removal of the nitrogen from the amino acid. So, as I mentioned, removal of the nitrogen from amino acid is the obligatory step during the metabolism of the amino acid. Sometimes removal of the, I will try to draw for a newcoming student. For a newcoming student, this is a general structure of the amino acids, okay? Then we remove amine group from amino acids, the remaining structure is known as a carbon skeleton, okay? I'm going to talk about this way, how we can remove the amine group, okay? We have it several ways. As you can see here, yesterday I introduced some transaminase enzyme. Do you remember aspartate aminotransferase, alanine aminotransferase? So these two enzymes are very, very important. Transamination, as you know, this is a conversion between two amino acids. For example, aspartate produces alpha keto acids. And all the time, we should consider this point, all the time, whenever we have a transamination, alpha ketoglutarate is participate, and alpha ketoglutarate following the intake of this amino group convert to the glutamate. So all the time in a transamination, alpha ketoglutarate and glutamate are participate. But what is the difference? Sometimes alanine is a donor of the amine group. It donates its amine group to the alpha ketoglutarate. So the name of enzyme is aspartate aminotransferase. But sometimes the donor of the amine group is, for example, alanine. When we say alanine aminotransferase, the donor is a amine. So glutamate is the amino group donor for the biosynthesis of non-essential amino acid. It means that whenever you add this amine group, at first you remove the amine group, okay, by the transamination. Is it actually result in a liberation of the free ammonia? No. We have another way. What happened? It's transferred to another compound. What is the name of compound? Alpha ketoglutarate. So, combination of the amine and alpha ketoglutarate is glutamate. Excellent. So, as you can see here, inside the glutamate we have the amine group. Okay? As I mentioned there, this amino group, because we have some essential amino acids. These are the non-essential amino acids. It means that our body can produce this amino acid. So if I want to synthesize this amino acid, I have to donate ammonia to them. For example, this is a precursor, and I want to convert it to the non-essential amino acid. This precursor requires amino group. What is the source of this amino group? Glutamate. So glutamate provides the amine group to the precursor that they want to convert to the non-essential amino acid. So this is the importance of this pathway. And sometimes glutamate has another fate. sometimes it's dominating amine group, and sometimes it participates in a glutamate dehydrogenase reaction. And in the next slide, I will introduce this important reaction. But sometimes there is no transferation, you know? For example, this is the amino acid, okay? This is an amino acid, okay? And this is an alpha keto acid, like an alpha ketoglutarate. When it transfers, this amine does not liberate in a free form. It binds to the another compound. There is no free liberation. But sometimes we have a deamination. When we say D, what does it mean?
Take away.
Yes, it separates and removes. For example, this is the amino acid and this is a deamination. So in this procedure, our amino acid produces free ammonia, okay? It doesn't transfer to another compound. It actually releases freely, okay? So this is a deamination. And most of the time, for example, serine and threonine remove their amine group by the deamination. But most of the amino acid, they remove their amine group by the transamination. So we have 20 naturally occurring amino acids. Most of them, 80 amino acids, remove their amine group by the transamination, but we have two exceptions like this. They release in a free form. Which amino acid? Serine and threonine. So this is a transamination. I think yesterday I will describe. I described it. This is the first step of the catabolism, aminotransferase. What was the cofactors and prosthetic group? What does it mean, prosthetic group?
It means it needs to attach. I think it needs to attach to the enzyme for it to be recognized, for it to be
to fully function.
Yes.
Yes. Do you remember about the coenzyme? We have two different coenzymes. One of them is a co-substrate that they loosely connect, and one of them is a prosthetic. Please pronounce by emphasize. Prosthetic. Prosthetic means very strongly bind. Okay? Whenever you observe the name of prosthetic, it means that it's attached to the enzyme by covalent bond. Okay? Prosthetic is tightly by the covalent bond. But sometimes we have some loose. For example, we have some enzyme that they require for example, folic acid. Folic acid loosely bind to the enzyme, so known as a co-substrate. So we have two types of coenzyme: prosthetic, tightly bound. Co-substrate, they loosely bind. Okay. So folic acid is the example of co-substrate because it's loosely bind to the enzyme. But about, for example, pyridoxal, it's tightly bound. So it is a prosthetic or coenzyme. So this is a vitamin B6, and all amino acids except threonine and lysine can participate in a transamination, and they are specific. And the name of the enzyme, as I mentioned, is related to the donor of the amine group. So about aspartate amino acid, what is the donor? Aspartate aminotransferase. The first name is a donor of the amine group. What is the donor? Aspartate. What about the alanine aminotransferase? Alanine is the donor of the amine group.
In the previous slide, you've written all amino acids except threonine and lysine can be transaminated, but before that we read serine and threonine.
Here.
But
We have a serine and threonine. Actually, because I take it is this slide from the another book. In some references, they mention serine and threonine. Actually, I put intentionally in this slides because maybe you encounter with other reference. In some reference, they mention serine and threonine just participate, and sometimes they mention threonine and lysine. Okay, so lysine is a debatable actually aspect of the catabolism of the amino acid. Why I put it intentionally, because sometimes maybe you encounter to this point. But for example, I didn't give you the name of the serine or lysine, but all the time you should know that you can take an threonine is the amino acid that do not participate in a transamination. But there is a controversial between lysine or maybe serine, okay? But all the time there is actually constant thinking about the threonine function. And this is a transamination. As you can see here, glutamate and alpha-ketoglutarate are the constant compounds that participate in a transamination reaction. And as you can see here, there is no free ammonia. We have transfer from one compound to another compound. So there is no free ammonia. And this is the alanine aminotransferase, aspartate aminotransferase, and pyridoxal phosphate is a coenzyme for this reaction. And you should know that glutamate, because participate all the time, it can act as a collector of the nitrogen. It means that it receives ammonia from maybe alanine, maybe from the aspartate, and finally they receive ammonia, and this is a collector of the ammonia. And you know, nitrogen itself is very toxic, okay? So normally we have a very low amount of ammonia, and especially for the central nervous system, okay? It's very, for example, I do apologize. Why physician advise the people, please do not hold the urine exertion.
Because of the nitrogen in the urine, if it gets
Because our
reabsorbed.
body gradually resorb the nitrogen, okay? One of the reasons is actually this point, because our body gradually resorb, okay? From the intestine, it's better to use this expression, absorb, okay? But about the kidney, it's better to use this expression, resorb, okay? The nitrogen, and they reach to the bloodstream, and finally they reach to the central nervous system. And the nitrogen, especially, is very dangerous for the brain, because it is very, very toxic. So how we can detoxify this nitrogen? What is the pathway to detoxify the nitrogen? Exertion as a urea, okay? So our body exerts urea into the urine to detoxify this nitrogen. And one of the sources of nitrogen in the urea cycle is an aspartate. And this is a deamination, non-oxidative deamination. As I mentioned, most of the references mention that serine and threonine. But some references mention that instead of serine, maybe lysine participate in a deamination. But you should know that following the deamination of the serine, what would be the product? Pyruvate and free ammonia. So there is no transpiration, okay? Please do not make a mistake. Deamination can result in the liberation of free ammonia, and transamination. Transamination all the time converts the amino acid to the alpha keto acid, okay? And produce a new amino acid. And also finally we have a propionic and succinate way. This slide is very, very important. Why? And at least one of you, your final question would be from this slide. Because this pathway is very important and desire special attention. Because glutamate is the very important molecule inside the body and can help us to detoxify the nitrogen. How? The sequential action of the transaminase, we have a sequential action of the transamination. Finally, they result in a transfer of the amino group from most of the amino acid to the alpha ketoglutarate. Okay, please start from this direction. I'm talking about this direction. Direction alpha-ketoglutarate toward the glutamate. So, as I mentioned about amino, I mentioned this one, most of the amino acid participates in a transamination. What would be the result of transamination? The production of the alpha-ketoglutarate. Okay? Whenever our body produces alpha-ketoglutarate, so how can remove this large amount of alpha-ketoglutarate and how we can dispose of nitrogen? So, sequential action, they produce alpha-ketoglutarate. And finally, this alpha-ketoglutarate collected free ammonia, okay? Because as a result of the catabolism, one of the end product of the many biomolecules is a free ammonia, okay? And during the transamination, we have a large amount of the alpha-ketoglutarate. So our body combines free ammonia with alpha-ketoglutarate and produce glutamate. So this direction is favored normally, okay? And by this way, it can provide a pathway that the amino group of the most amino acids can release as a ammonia, like here. And as I mentioned, because this ammonia is very toxic, it's combined with alpha-ketoglutarate that results from the transamination. And now we produce glutamate. Now glutamate is non-toxic. So alpha-ketoglutarate absorb all of this toxic molecule to produce a non-toxic molecule. And also please pay attention to the different type of coenzyme. Whenever our body wants to convert alpha-ketoglutarate to glutamate, which coenzyme is required? NADPH. What about the dehydrogenation of glutamate? NAD is required. And in this actually reaction occurs primarily in the mitochondria of the liver and the kidney. So these organs, liver and kidney, are vital to detoxify this dangerous molecule. And this free ammonia following the glutamate converted to the alpha-ketoglutarate is a source of another source of nitrogen. What was the first source of nitrogen in this slide? Aspartate. You know, in the urea structure, we have a carbonyl and two amine group. One of them results from aspartate, another one results from removal of the amine from the glutamate. And we have some activators like ADP. These are allosteric modulators like ADP activate this pathway, while GTP can inactivate and inhibit the function of these enzymes. About the transport of the ammonia from the peripheral tissue to the liver. You know, as I mentioned, one of the end product and byproduct, it's better to mention byproduct. One of the byproducts of many biomolecules is ammonia, NH3. But you know that ammonia is very toxic. How we can detoxify? According to many procedures. For example, in most of the tissue, like skeletal muscle, like liver, like CNS, following the metabolism, our body produces free ammonia. How we can exert finally into the urine? At first, we should start with the combination of the glutamate to the amine. Glutamate, please do not make a mistake. Alpha-ketoglutarate combined with the amine. What would be the result?
Glutarate.
Glutamate. Alpha-ketoglutarate. This is an alpha-keto acid, like this. This is the alpha-ketoglutarate, for example. Combined with the amine, what would be the result? Glutamate. Okay? This is alpha keto glutarate. Whenever you add amine, what would be the result?
Glutamate.
Glutamate. Okay? So, whenever, for example, glutamate, this is a side chain of glutamate. Am I right? Okay? But whenever you add one more amine to the glutamate plus another amine. For example, you add from here. What would be the result?
Glutamine.
Glutamine. Okay? Please do not confuse these two different amino acids. So, following the attraction of free ammonia from the metabolism of the most of the tissue, our body produces non-toxic transport of the ammonia. And because we have some system to transfer a glutamine, this glutamine can reach to the liver. Okay? From the tissue, it's sent to the bloodstream and finally reach to the liver. Liver is a specific and vital organ to detoxify this free ammonia. How? Again, this glutamine digested by the glutaminase. Okay? Then we remove it here. I'm talking about here. This is the glutamine. Okay? Then we remove amine group from glutamine. What would be the product?
Glutamate.
Glutamate, okay? So, glutamine is the non-toxic form for transfer of this toxic amine from other tissue to the liver. Inside the liver, our body removes the amine group and produces glutamate. So, this free ammonia, because you know, this is exactly like very bad-tempered children. You cannot release it freely, okay? You should hold his hand. This is ammonia, okay? You should hold as a glutamine and reach to the liver, and now you can release his or her hand, okay? So, finally, this ammonia by the cycle, in the next slide I will introduce it. Urea, this free ammonia finally convert to the non-toxic of the nitrogen and can exert into the urine. This part is not important, okay? You should just know that we have two ways actually. In most of the tissue, glutamate hold the hand of the NH3 and convert to the glutamine. Glutamine reach to the liver. Following the glutaminase reaction, our body, actually glutamine, liberate the free ammonia and finally by the several steps convert to the urea, and this urea reach to the kidney and exert into the urine. Another way for the disposal of the nitrogen is the glucose-alanine cycle. And you should know that alanine plays a pivotal role and a special role for transporting of the ammonia group. What happened here? This is the glycolysis. Do you remember the glycolysis pathway?
Yes.
No. This is the aerobic pathway. What was the end product of an anaerobic?
Lactate.
acid.
Alcohol and lactic acid.
Okay, for example, if you have an abnormal exercise and vigorous exercise, have you experienced?
Yeah.
You have to run very fast. Okay, you have to run very fast to reach, for example, to the taxi. Okay,
Then there is a pain.
there is a pain. Why?
Because of
Reformation of
lactate.
Yes, because you have a lack of oxygen, so glycolysis, aerobic glycolysis shift to the anaerobic glycolysis because you have a hypoxia. Okay, so as a result of this lack of oxygen, instead of pyruvate, our body produces lactate, so it can result in a pain. And what is the best way to actually remove a lactate?
To keep
Convert into
Some
pyruvate.
vitamin C and also tomato. Tomato can help us to exert the excess amount of lactate. For example, you are not very professional athlete, okay? For the first session of the exercise, most of the time, you cannot even sit. You know, have you ever experienced? You feel a lot of pain inside the muscles, okay? How we can control, one of the way is the taking of the vitamin C to dispose the excess amount of the lactic acid, okay? And some actually physician prescribe a tomato because tomato can combine with the lactic acid, can exert the excess lactic acid. Okay.
Gets from its hand.
So, glucose during the aerobic glycolysis converts to the pyruvate. And what is the reaction, conversion of pyruvate to alanine? What is this reaction? Conversion of pyruvate to alanine.
Again?
Transamination.
Yes, this is another direction. All the time I mention alanine to pyruvate, alanine aminotransferase. This is the reverse, okay, direction. So by the aminotransferase alanine, you know, because we don't have a specific transport for pyruvate, our body have to convert the pyruvate to the alanine. Now this alanine, for example, have this permission. For example, this is a barrier of the cells, okay? Now I'm a pyruvate. There is no permission to me to pass through, okay? So how can I pass through from the, for example, muscles? I have to convert, for example, to alanine, okay? After taking amino group, now I have this permission. And I reach here, okay? Then I reach to the liver, again by the transamination, I liberate my pre-ammonia, and now again I'm pyruvate. So this is another way, and this is a cycle. And this pyruvate by the gluconeogenesis convert to the glucose. And by this reaction, glutamate produced, and this glutamate, according to the glutamate dehydrogenase, liberates the ammonia. Do you remember this reaction?
Yes.
This is the very important slide. Whenever alanine, okay, remove, convert to the pyruvate. What happened to the another part of the reaction? Alpha ketoglutarate convert to the glutamate. So it means that I transfer my amine group to the alpha ketoglutarate. And this alpha ketoglutarate by the glutamate dehydrogenation liberated free ammonia. And this free ammonia finally inside the liver can form a urea. What is the urea cycle? Have you ever heard this cycle name?
Yes.
It participates inside the liver because we have it, I will show. We have one specific enzyme, and because of that specific enzyme, we cannot actually produce a urea other side of the body. So liver is a vital organ for the disposal of the nitrogen. And this is the major disposal form of the ammonia group. And as you can see here, this is a structure, this is the structure of the urea. How many nitrogen atoms is present? 2. So, if you remember, we have a source of nitrogen. One of them is provided by the oxidative deamination from that important slide. Glutamate convert to the alpha ketoglutarate and liberate free ammonia. This free ammonia participates in the structure of the urea. And what is the another source of ammonia? Aspartate from the amino transferase reaction. And what is the source of CO? CO2. So the source of this nitrogen and oxygen and carbon is important. So urea is produced by the liver and transported to the bloodstream to extract from the urine, from the kidney. This is a urea cycle. As you can see here, this is a cycle. Remembering the details of the cycle is very important. Okay, Bismillah. CO condense with the free ammonia that results from the glutamate dehydrogenase most of the time. Okay? CO, whenever combined by the help of how many ATP? 2 ATP. Convert to the which molecule? Carbamyl phosphate. What is the name of enzyme? Carbamyl phosphate. Synthesize 1. Because we have a number 2 in some nucleotide biosynthesis, so number 1 participates in this reaction. Please consider the location. The number 1 reaction takes place inside the mitochondrial matrix. Ornithine transfer inside the mitochondria and combine with carbamyl phosphate with the ornithine transcarbamylase and produce citrulline. Ornithine and citrulline are an example of basic amino acid, but they do not participate in a structure of the protein because we have only 20 amino acids. I don't know, valine, leucine, isoleucine, aspartic acid, glutamic acid. These are amino acids, but they do not participate in the structure of amino acid. They have another role, for example, like in a urea cycle. Okay, what happened to citrulline? It's sent outside of the mitochondria and produces arginosuccinate. Please attention to this ATP. How many phosphor high energy bond is here? How many ATP is used? 2? Here. Only 1. But how many high energy bonds used? 2. 2. Why? Because instead of ADP, our body produces AMP. Okay? So, if I ask you about the next slide, I will show the stoichiometry of the urea cycle. It's better to show there. Please consider 2 ATP used here to produce the 2 ADP and 1 ATP converted to the AMP. What happens to the arginosuccinate? By the arginosuccinase, it's very clear. It can digest to the arginine and fumarate. About arginase enzyme, as I mentioned, this is a specific enzyme that can only and exclusively found inside the liver. Because of the lack of this enzyme in other parts of the body, our body cannot produce the urea. For example, in a muscle, I don't know, in a kidney, in a bone, because we don't have any arginase enzyme, we cannot produce the urea. So if I ask you, which of the following enzyme of the urea cycle is specific for this cycle and it's specific for the liver, we should choose the arginase. And finally, by the digestion of arginase, ornithine again recycled and urea exerted into the urine. And the first enzyme is very important, and we have activator for this enzyme, and the name of activator is N-acetyl glutamate. This is the overall stoichiometry of the urea cycle, aspartate, free ammonia, and CO2. And how many ATP is required? So maybe I ask you, for the complete urea cycle, how many ATP is required? 3. So maybe I ask you, for the complete urea cycle, how many high energy phosphate bonds?
4.
Excellent. So only 3 amino acids, 4 high energy is required. And this is a review of the source of ammonia. And for example, a glutamine can result in a catabolism of amino acid, especially branching amino acid like valine, leucine, and isoleucine. They can produce the glutamine, and they're taken up by the intestine, kidney, and the liver. This is a review. In the intestine, we have, for example, glutaminase. They liberate a free ammonia. In the kidney and liver, we have a glutaminase and also dehydrogenase to produce the free ammonia. And this free ammonia can detoxify in the urea cycle and form urea. And sometimes, please pay attention, sometimes this is a glutamine, okay, am I right? This is a glutamine. Convert to the glutamic acid and free ammonia, okay? Sometimes it detoxifies as a urea in a urea cycle, and sometimes, especially in a kidney, it produces, instead of ammonia, it produces ammonium, okay? It means that it takes up the proton. What do you think about the role of this pathway? When this pathway, combination of the proton with the ammonia and produce ammonium is important. What do you think about the importance of pathway? Why sometimes our body, it's better to explain as at this point. Why our body do not use the total amount of ammonia for the biosynthesis of the urea? Why a small part of ammonia combined with the proton and produce the ammonium? What is the importance of this way? What do you think?
Because of H positive, there should be more acidity, pH increase. So,
Merci.
so ammonium is less toxic, so it buffers the pH.
Excellent.
Yeah.
Sometimes we have an acidic environment, like acidosis, okay? Actually, we have abnormal amount of proton. Whenever you have an abnormal amount of proton, it means that the level of the pH is low. So, as a buffering and a balance of acid and base amount, our body wants to remove this proton. And one of them is a production of the ammonium. So this way is very important for the balance of acid and base. And also some of the other sources, for example, we have some bacteria can act as a source of the production of the ammonia inside the intestine, and sometimes we have some amides that obtained from the diet, and catabolism of the nucleobase like purine and pyrimidine can result in ammonia. And sometimes we have some metabolic disease about these cycles. Sometimes some of these enzymes, like enzyme number 1 and number 2, are deficient. And most of the time, they can result in a hyperammonemia. It means that we have a high concentration of ammonia inside the blood. Excellent. And as I mentioned, these are the disorders. What would be the result of the accumulation of the ammonia?
Poisoning.
Poisoning, especially for
Brain.
brain and central nervous system. So you expect that most of the manifestations are related to the CNS, central nervous system. So we have different, some of them is acquired. For example, you have a liver surgery. You have an inflammation of the liver, because as I mentioned, the site of the urea cycle inside the liver, any damage to the liver can result in a hyperammonia. And sometimes you have a normal liver, but you have a genetic disorder, especially in some enzyme that catalyze the reaction of the cycles. And what is the important deficiency? Ornithine transcarbamylase. So if I ask you, which of the enzyme of the urea cycle is more susceptible to be deficient in the urea cycle and can produce a metabolic disorder, is the enzyme ornithine transcarbamylase. And as I mentioned, most of the symptoms like tremors, like slurring of speech, they don't have any control to their words, and they have drowsiness, vomiting, and other actually blurring of the region. And all of them are as a result of accumulation of the ammonia. And in a very severe situation, for example, like this, I encounter with these patients. You know, they don't have any control, they cannot pronounce very clearly, like this. And finally, as an accumulation of the high amount of ammonia, because it, as I mentioned, this is very dangerous for the brain, it can result in a coma and death. And how we can distinguish according to the blood, urea, nitrogen, BUN level, high ammonia in the blood, and elevated glutamine. And also other metabolites or accumulate according to the which enzyme is defective. So how we can treat? The best way is the restriction of the protein, because the main source of the ammonia is protein. For example, it's better to change in a vegetarian, okay? So you can control by the low protein diet, and you can administrate some compounds that bind covalently by the, for example, some metabolites, and they can exert that metabolite inside the urine. For example, treatment of the patients who are who have a defective of the urea cycle enzyme, you can administer them by the phenylbarbiturate. This phenylbarbiturate accumulate for the some toxic compounds and aid to exertion of the ammonia. Just the name of the phenylbarbiturate, the mechanism is not important because they accumulate by the many precursors, many compounds like free ammonia, like glutamine, and they can help them to exert in the urine. So this is a summary of the amino acid metabolism. So in this session, I describe what happened to the protein. The protein digests to the amino acid. Amino acid absorbed to the intestine. And then at the first step, the amine group removed. Okay? Maybe it's removed by the transamination or by the deamination. We have a threonine by the deamination, but some references they mention that maybe serine or lysine. Okay? So, and as I mentioned, what happened to the free ammonia? It converted to the urea. In which organ? Liver. What was the name of cycle? Urea cycle. So we have a detoxification process of this free ammonia inside the liver. So what was the remaining structure of the amino acid? After removing of the amine group, carbon skeleton. So for the next session, I will describe what happened to this carbon skeleton. Okay? I think at 10 o'clock. Okay? I will start at 10 o'clock.